Thank you. Okay, so I'm gonna talk about this project. I already presented this in the on-site meeting we had here in San Diego, but this can be like an update of what we have done. We didn't do significant advances on this, but we'll try to anyway summarize everything and give you an update. So let's see. Okay, the line is going to be this. I'm going to start with an introduction of why we are interested in protein structures and which links are between the protein structures changes and diseases. Then I will show our experimental workflow and then how we compare the experimental data we got from there to the PDB structures, the crystal structures that are deposited in PDB. Then I will show further developments like some improvements in our experimental workflow that will allow us to get more data and more flexible way, and then some next steps in our developments. So the structural proteomics is very interesting and is, as you may already know, is involved in many diseases. The alteration of the protein structure can lead the change in protein-protein interactions and therefore change or cause different type of diseases like infections, proteopathies, like aggregation of protein aggregation, like diseases like aging, Alzheimer, amyloidosis, other diseases like misfolding, like cystic fibrosis, where we have a lot of expertise, or others like tumorigenesis. So to know which cause the alteration of the protein structure can lead us to know to have more insight on these diseases. So in general, in a standard proteomics experiment, we identify and we quantify proteins. That is shown here, where we usually get a distribution of ratios, and then we apply some statistics to see which proteins are significantly regulated between the two samples that we compare. But here, our aim is to determine a quality of a protein. So we want to quantify in a proteome-wide and in vivo how the protein folds and how the protein interact with each other, looking to the quality, which is the accessibility of certain sites in the sequence of the proteins. For this, the experimental method is summarized here. We have our cells in vivo, and there we can have the proteins in different conformations. So the idea is that we are going to measure what is the proportion between, for a certain protein, which is the proportion between the conformation that is accessible and the conformation which is not accessible, looking to a particular site. Okay, so for that we first label in vivo, and therefore the sites that are accessible will be labeled, and the ones that are not accessible they will not be labeled. Then we perform enzymatic digestions, and then we perform a second labeling and therefore the sites that were already labeled in vivo, because they were accessible, they are not labeled anymore, but the ones that were not labeled because they were not accessible, they are labeled now. So at the end, after the mass spectrometry analysis, we are going to have some ratios between the two labels, and these ratios are going to give us some measurement of the relative surface accessibility for those sites. So this is one example of one experiment in which we analyze HEC293 cells. We were able to quantify almost 8,000 peptides, in which, as you can see here, at the right, the distribution of the ratios. Most of the ratios were negative, meaning that more than the 93% of the lysines were accessible in vivo, which is what we expect. But you can see here, there is a population which is having lysines that were not accessible in vivo. Here, the CTRP1, that is serves as like a positive control, because that is the chymotrypsin, and is expected to be only labeled with the second label, so that is why it's inaccessible there. And therefore we focus our analysis then in these 155 significantly inaccessible lysines residues with a particular false discovery rate of 1%. And then analyzing these proteins having these inaccessible lysines, we prove that there is a significant enrichment on these protein classes, like translational elongation factors, translation initiation factors, ligase, RNA binding proteins. So we see that are proteins that are interacting and therefore is expected. It confirms us that these sites are functional units or are part of functional units of links between these proteins and other proteins or molecules. These are with the representation that we have with the protein cluster quant. We have some of these cases in which we see these nodes more in yellow, that are the lysines that are inaccessible, and they are pointing us to some isoforms of these translation initiation factors that are that have these lysines inaccessible in vivo. I don't have the images here, but then we map these sites with the 3D structural data that is there, but I will show you now more examples. So then, having this experimental data about the in vivo accessibility of these sites, we wanted to compare with the available 3D protein structures in PDB to see if the structure in vivo is concordant to the data that we have. So then the question is, which are the 3D structures that best match the experimental data that we have? So for that, there is a first step in which we have to map the quantified sites to the sites in the structure. And this is not trivial because first we have to map our quantified peptides to protein site. That's fine. So we have several peptides that are overlapping in the sequence, and we have a particular ratio per peptide. So we have to pull all these ratios and assign them to particular sites in the sequence, in this case of proteins with the sequence in UniProt. And then we have to map these proteins to PDB entries. So for example, these two peptides, this is a mock example. Let's say that we have this site 125 in the protein P12345 in UniProt, and then we map that to PDB, and we say we have these three structures for that protein. Then for that particular site, we had to map it to the sequences in the structures. And therefore, we can have, for example, that this site, the site 125 in this protein corresponds to the site 155 in the chain A of this particular structure, or the site 153 in the chain B in the structure. So now you are already realizing that the mapping is not one-to-one. So we have to deal with this the heterogeneity of data in the structure coming from the UniProt. And then when we have the sites recognized in the structure, we have to calculate the surface accessibilities for that structures. So also we have another problem that for this particular experimental workflow, we have just one measurement per peptide, meaning that this measurement is coming from the license that the peptide have. But sometimes these peptides have more than one license, so we have to just take the ones that are coming from one, only to take the peptide that contains only one license, because as you can see here, the peptides in white, we don't know this measurement in which proportion each site, each lysine is contributing to the final ratio that we have. So we have to filter the peptide in that sense, and then have one accessibility ratio per protein site. Okay, and then in the second step, so to map these proteins to the PDB structures, so we realize for this previous experiment that in average, each protein maps to 17 PDB structures, but some of them, they match to 100. So it depends on how this protein was studied. We can have many structures in PDB. We assume that with obviously certain similarities, but some of them, they belong to different conformations and so on. And in average, we already realize that each protein site is present in 28 different models. So the correspondence is between the UniProt sites and the sites of a particular chain in a PD model is not one-to-one, it's one-to-many. Then the last step is to map the sites to the sites in the 3D structures. And then sometimes we have the problem that the sequences doesn't match. So we don't have that particular peptide in the PDB structure. And that is because of many reasons. Sometimes it's because the protein it was mutated, or is a particular subset of the sequence of the protein, is a partial protein in the structure. So we have to discard those cases also for this comparison. And finally, when we have the mapping to the particular sites in the structures, we use a Gmol set of commands to calculate the solvent accessible surface for a particular atom in the amino acid in the sites that we mapped, and then we get a value. a value that is a percentage, and as you can see here, this is a visualization of the 3D structures. So we have a value here of around 59%, meaning that that is the percentage of the sphere that this particular atom have accessible to the exterior of the molecule. So, but then, as I already mentioned, the PDB structures, the entries usually contains more than just the protein. So they can contain multiple chains when they are part of a polymer, or they also contain solvent molecules, ligands, DNA, RNA. So it depends on the case. There is much more information there, and therefore we are not sure which is the in vivo context in which our proteins are. So we have to see which structure is the more similar to what we have. So yeah, depending on if we take into account other molecules or other chains in the structure, we're going to have different surface accessibilities. But in average, we saw in this case that taking all the sites that we quantify, the difference between removing all other things that are not our protein and having whatever it is there is just about the 2% in the value. But there are extreme cases like this one. So for example, for this protein, we by default having all the molecules in this structure, we have a value of 2.58% of accessibility. You see here the red atom here is the one that we are measuring, and you see there are a yellow chain here, and then these green molecules here. So these green molecules are glycerol. In this case, this structure is including that, and you can see here more in detail. And then if we decide to remove this glycerol, we have a much different value at 20%. But then you can see here the difference in this orange surface is the surface. And then if we say, okay, let's take just one monomer of this protein, just one chain of this structure, which is our value now. So taking just the white one is going to be 58.98. So there is these cases in which is very different what is available to the surface or not in the structures. So okay, we have to deal with that, but we have to take into account that in vivo we also have these cases. I mean, depending on what we have, on how the protein is, which is the conformation of the protein in vivo, we have these differences, and we are going to have these in our final ratios is a mix of these populations or different conformation of the proteins. So then we compare here, taking the most the closer surface accessibility to what we have. We have this distribution of accessibilities. So in the y-axis we have the 3D structure accessibilities, and in the x-axis we have the experimental ones. So there is some correlation, positive correlation, but still there are many cases in which we have differences. And the idea is that we want to explore these cases more in detail. So in summary, we have experimental information that not always match the PDB structure. So we don't know which is the minimum surface required for a lysine to be labeled. So that's also interesting to know. And then multiple different conformation are contributing to our experimental ratio. So we have to take into account all these cases. So for some cases, it is worth to explore which is the 3D structure that best explains the experimental data. And we developed this tool for automating the comparison of this data, experimental data, and 3D structured data. This is the next step. So it's an improvement in the experimental workflow, which is based in the addition of additional labeling. So we have before I was showing this labeling, which it was based on MS1 labeling, but now we have an additional labeling, which is the isobaric isotopolog labeling. So we have two samples and they are labeled. Sorry. Okay, I guess okay. So we have these two samples that are labeled with this first labeling, and then we have the MS2 level labeling, the isobaric isotopolog, which is the one giving us the surface accessibility information, and the previous one is going to give us the difference in the expression of this between two samples. Okay, so we have two levels of information here: regular or common quantitative information in the first labeling, and the surface accessibility in the second labeling. So we have data for this approach, and we saw that we increased a lot the dynamic range. So you can see at the right the distribution of ratios. You can think at the beginning, seeing this chart, that actually the ratios are compressed, but what is happening is that we have a lot of data in the extremes. So we have ratios of accessibility that are infinity here and that are minus infinity here, meaning more accessible and less accessible, completely accessible and completely inaccessible. So because in the other case the distribution it was more we didn't have these cases because it was basically in Silac, and therefore we have always a signal of noise, which is making our ratios having some particular values, but not these extreme ratios. In this case, because it's MS2, we remove all these noise, and we can have these infinity cases in log 2. I didn't say that. It's in log 2, so we can be more sure about the values that we are having. And this technique has a high reproducibility and low sample throughput. But we are having this information already, and we are integrating this in our comparison. Also, because of this isobaric isotopological labeling, we can have information about these cases that we were discarding before. So peptides having two lysines with the information of the surface accessibility. So we were discarding them because we don't know which is the proportion for each side contributing to the final ratio. But now we have MS2 ratios, meaning that we have a ratio per series of the MS2 peaks. So there are some cases in which, if we analyze properly the data, we are going to have information about the sites separately, so we can distinguish each site in a single peptide. So as a future, the last part of the talk is the future perspective of this project, and is to integrate this tool with other resources. You already know the protein cluster 1, PCQ, in which we have this cytoscape visualization of the data, and that is pinpointing some cases of proteoforms that are significantly regulated. So the idea is to integrate this surface accessibility in PCQ in the sense of, instead of aggregating all the peptides that share the same set of proteins, and the same for the proteins, aggregate the proteins sharing the same set of peptides. So instead of that, we want to aggregate or merge the peptides in peptide nodes by the sites of the proteins that are quantified. So in this case, the peptide nodes will be protein sites, particular sites, instead of peptides. Okay, so this will help us in this type of experiments to identify data inconsistencies due to shared peptides, and then also to integrate multiple experimental ratios for the same site into one single value, which is going to allow us to perform outlier removal statistics, and therefore we're going to have more reliable data. Also, we plan to integrate this in Paint. So the idea is that the people goes to Paint, which is this online database for proteomics experiments. So the people can go there, select a particular peptide from an experiment, and then show the valuable PDB structures, and then to visualize this particular site together with the surface accessibility, something like this scheme. So because we already have this code, it's going to be easy to integrate in our online tool. And then we can think about having apply this to particular cases of interesting PTMs in peptides, but also for these surface accessibility experiments, we can then generate plots of the comparison between the experimental accessibilities and the structure accessibilities or something like that. In conclusion, we have quantitative measurements of local protein surface accessibilities, and the novelty of this is that it's done in a proteome-wide fashion, and it's done in vivo. Then we map the experimental measurements to 3D structural derived surface accessibilities in PDB, and this comparison lead us to realize that in vivo experimental data is not always reflecting the structures that are in PDB, and that our tool helps to choose which is the closest structure to the experimental data. Obviously, can also help to characterize new structures for other proteins that are not available. And this, and we are planning. I saw that we are planning to integrate this resource in other tools like PCQ to get more accurate data, and into Paint to get accessibility measurements for particular sites and visualize them online. Just to thank to Casimir and Sunny, which are the ones making the experimental part of this project, Robin for the support in all these analysis, and obviously John and the J's lab team. So now if you have any question, I'm happy to answer. Thank you.
Thanks, Salvador. Are there any questions? Okay, if there aren't any questions, thank you, everyone. Thank you, Salvador, for the great talk.
Thank you.
This concludes our conference. Thank you, everyone, for attending.
Thank you very much. See you.
See you. Bye.